Hello, welcome to Winter Rosa Apiaries.
Time sitting down and listening. So, I'll try to make this interesting and really happy to take questions throughout or afterwards if you're interested in it. I'm going to tell you a little bit about honeybee viruses today. And the sort of first thing to note about honeybee viruses is we have a lot of gaps in terms of the research and our understanding of how viruses work. But I'm going to try to give you a sense of the background, what viruses are, how they work, and also talk about symptomology and some things we can do to improve colony performance despite viruses. So we have this weird little board. Okay. So I wanted to start out by just reminding you that colony loss, whether that's CCD, colony collapse disorder, or just colony mortality without CCD symptoms, it's pretty well accepted at this point that this is a result of multiple interacting stressors. So a bunch of things interacting. They converge on the individual and the hive. So a single bee may have a lot of different stressors it's facing, but then the hive as a whole will also be having different individuals impacted by different stressors. So one of the major components of this kind of stress framework is the role of pathogens. And so you've probably heard a lot about varroa mites, that's a parasite, food nutritional stress, pesticides, these are other factors that come into play. Pathogens come in a few different forms. These are just really disease-causing microorganisms. Bacteria can cause disease, so something like American foul brood, that's a bacteria. Funguses can also cause disease, so nosema is a fungal pathogen. Viruses are a specific type of microorganism that cause disease, so these are viral pathogens. So I'm only focusing on really one category here of pathogen. These designations are important. For example, bacteria are treated with antibiotics, viruses aren't. So it's important to know something about these distinctions. You're all familiar with viruses. We deal with them as humans. So Zika is probably one that's captured your attention in the last few years. There's also the common cold is a good example of a virus that we have to deal with. So what is a virus? These are weird little creatures, and I think it's important and maybe helpful to think about the biology a little bit. So what this is showing is if you were to look under a microscope and draw a cartoon of a virus, this is what viruses look like. There's a lot of variation and diversity. So these are just some common viruses, HIV, influenza, or flu. Bacteriophage is a virus that actually acts on bacteria. So even bacteria have to deal with viruses, they have to contend with them. So they look kind of weird. Maybe they look a little bit like a cell or something like that. What's going on here? Well, what's interesting about viruses is they're not actually a cell themselves, they're a cell parasite, which means that they infect the cell and take advantage of all of the machinery inside the cell to make new viruses. And the reason this is important is because it gives you some idea of what it is that actually causes symptoms when you're infected with a virus. So I wanted to tell you a little bit about the structure here, and there isn't going to be a quiz, but just try to think a little bit about how this works. So a virus is very simple. It basically contains a genome, so similar to us, it has some form of either DNA or another related molecule that carries all of its genetic information. So that's what's going on inside here, or this is another example. And then it's got a protein covering on the outside called a capsid. That's pretty much it. Those are the nuts and bolts of the virus. So it's this little kind of protein floating around that has genetic material inside. It can't replicate or reproduce outside of a living cell. It has to have the machinery in that cell to replicate. And so the cell is providing the machinery basically just to make new viruses. So the life cycle or strategy of a virus looks very complicated, right? This is a cell, like a cell you have in your tissues, let's say a skin cell. These are viruses that are in their little capsids attacking the cell, going inside. But basically all they're doing is using this to make more virus. That's their whole lives, right? They're just replicating and spreading their genes. It's a very simple outcome. The consequences for the cell are varied and can be pretty negative. virus is, how does it make you sick or a bee sick? Basically, there are various types of processes that cause symptoms when you're infected with the virus. Usually, these symptoms are caused by the death or damage of the cell the virus is inside. Pretty intuitive. And just for terminology's sake, this is called pathogenesis. What is it that's actually making you sick? And just as a sort of spoiler alert here, in a lot of cases in bees, we don't actually know why it is that the virus is making the bee sick, what is causing the symptoms. We know that there is a relationship between having the virus and being sick, but what is in between is unknown. So that's a big area of research that is sort of, waiting to be discovered. So I wanted to give you a simple example of a virus that you're at least familiar with, hopefully you've never experienced, which is rabies. So why does rabies make you sick? That's because when you get a bite from an animal that's infected, the saliva has the virus in it. It gets into the muscle and eventually into the nervous system. So rabies goes right into the neurons, the cells that make up your nervous system in your brain. It replicates in those cells, and then as the virus replicates, it spreads towards your brain. It goes from your peripheral nervous system into the central brain, and that's where it causes the most damage. So as it moves through these cells, it causes nervous system dysfunction, ultimately leading to these crazy behaviors we associate with rabies victim. So it makes people or it makes animals aggressive and crazy because it's infecting their nervous system, right? Right. So I said this before, but the mechanisms that cause these symptoms. So, for example, this is worked out to a degree with rabies. Actually, not to the detail you would expect, but at least we know where is it infecting the individual, why is it causing behavioral symptoms. We know that for rabies, we don't know that for bees. So even which tissues of the bee are targeted by a virus is in many cases not known. Okay, but just thinking about the sort of general knowledge of bee viruses, how many are there? That's seems like a relatively simple question. For example, we can ask how many bees are there in Kentucky, bee species. But that's actually pretty tricky to answer as a question. And this is another reason that viruses are so powerful. So remember, a virus's job is to replicate and make more virus. And this process of replication can lead to mutations in the genome of the virus. As a result, if you look inside, let's say, a honeybee, it's not necessarily infected with one type of a given virus. There might be a lot of different variants of that virus that are sort of replicating and changing as it's living inside the bee. So there's some interesting features of this. So for example, certain viruses can compete with one another inside an individual bee, and one may dominate, and that's the one that will be spread to other bees. So that's sometimes called a master sequence. And so one of the things that's becoming interesting, I'll talk about specific viruses in a second, but there's some work refining, which types of viruses make up something like the formalin virus. There are actually several different strains, and strains have different symptoms. That's why that's important. So I put this up here. This was last week's news or the week before. Studies linking multiple sclerosis in people to certain variants of the herpes virus. Not every strain, but certain strains. So similar to that, you can get very specific disease symptoms associated with one strain of a virus and not the other. So one of the things I'm trying to do here is sort of paint a picture of complexity. We're trying to understand how viruses affect bees. Okay, so just to give you a little bit of a virus survey, most bee viruses fall into a particular sort of family or group of viruses called Picornaviriales. This is a weird word, but if you break it down, all it means is pico, small, and then this virus's genome is not made of DNA, it's made of its cousin, RNA. Not important to know for the sake of this talk, but just to give you a sense. So there are other types of viruses that infect animals, cattle, bird viruses, also humans, that are in the same category, right? So these viruses are actually related across different hosts, which is an interesting feature of viruses. But that's one of the reasons that they're able to infect hosts of a variety of species. So here's a list of most of the these picornaviruses that are in honeybees, and I've colored these to give you an idea of the ones that are kind of related to each other. So we have various types of bee paralysis viruses that are related. Deformed wing virus and Kikugo virus, Varroa destructor virus. These are actually related to one another in terms of the sequence and the function. And some others, sac virus, slow bee paralysis virus. There are the Lake Sinai viruses that are sort of the outliers of these groups. So in my opinion, thinking about viruses, it's not necessarily important to memorize all of these and what they do. They actually share some symptomology. But just to give you an idea that, like as in humans, there's a lot of variation here. It's diverse, and understanding which of these viruses are most destructive and how they're acting really requires knowledge of some of this variation. Okay. So when I think about bee viruses and talk about them, I actually describe them pretty generally. A lot of these viruses have very similar symptoms. So from the standpoint of a beekeeper, sometimes it's useful to know which viruses your hives have. Other times you can just identify the symptoms and still treat that colony, treat it for those symptoms. Okay, so remember how these symptoms are caused are still largely unknown, which is interesting. But paralysis viruses all have some similar symptoms: trembling body and wings, an inability to fly, this kind of like hairless and greasy look that you can see in these bees here. Deformed wings is, of course, a telltale sign of deformed wing virus, and a lot of you have probably seen that. That's a very common virus that's vectored by varroa mites. This is a picture of black queen cell virus and sac brood virus, which are more rare. Okay, so in general, I think it's useful to just know that these are viral symptoms. Memorizing the connection to a particular virus may or may not be very helpful at this stage. Right, so these are individual symptoms. This is another important thing to keep in mind with honeybees. Colony functions as a unit, right? So you have individuals that are infected, but you also have colony level consequences. So many of these viruses have been associated with overwintering loss, just that your colony dies over the winter at a higher probability if you have that virus. Others are associated with reduced worker populations, meaning smaller numbers of worker bees. That will make your colony less productive. Okay, one of the things that I think is important to note with honeybee viruses, and this comes back to the importance of varroa mite control, is that there are different transmission routes to viruses. Different individuals get exposed, and the viruses can be transmitted in different ways, and that can often impact the symptoms of the virus. So there are kind of two major types of transmission when we think about any virus and any organism, really. Vertical transmission, so top down, that's when a parent infects an offspring. And then horizontal transmission across. So this would be, in our case, transmission among individual worker bees. And this horizontal transmission is typically the type of transmission we're concerned with honeybee viruses. Okay, so this slide is showing you the variety of ways that bees can actually acquire viruses. And here, this is not necessarily virus specific, although there are some details there, but I'm not going to go into those. So these are the general transmission routes. B2B feeding, that might be intuitive. Direct body contact, bees are very much in physical contact with one another all the time. That can actually lead to viral transmission. Contact with bee feces, this is a picture of a hive entrance that was probably infected with nosema, so there's a lot of feces around. But that's a good way to transmit viruses as well. There is evidence of queen to offspring virus transmission, but that's probably not as important as some of the other modes. And then varroa feeding is probably one that you've heard about, so varroa actually biting the bee and then injecting the virus. Okay. Varroa mites are pretty substantial contributors to viral transmission, but that's not the only thing that they seem to be good good at in terms of this interaction with viruses. So I'll go into some detail here. Varroa mites are known to transmit at least 5 different viruses. So they're moving a lot of viruses around from bee to bee. Deformed wing virus is probably the most iconic virus associated with Varroa mite. There's a lot of studies to show that they transmit deformed wing virus, and that feeding by Varroa mite impacts the replication rate of this virus. So that means that if you're a bee and you're infected, that virus is going to perform better and be more likely to cause symptoms if you're also being fed on by Varroa mites. So this is where symptom management is an issue. Viral presence itself may not be that big of a deal if you don't have other stressors that impact replication. So that's where symptom management becomes about minimizing stress. So Varroa is a key stressor to consider here. This figure I just wanted to show you because it gives you an idea of how the quantity of a virus or the abundance actually impacts symptoms. So this is a measure of deformed wing virus quantity inside a bee. And then there are different groups here. So we have uninfested bees that aren't deformed, so they seem asymptomatic. They may have low titers of deformed wing virus. It's not 0, as you can tell here. Much lower, but not 0. Infested, non-deformed bees, so these are bees that are infested with Varroa mites, they still have a fairly low deformed wing virus titer. So they're not showing these wing deformations, meaning they're not showing the symptoms. So this is providing evidence that mites are actually collecting this virus and transferring it. So if you look at the mites themselves, they have a fair amount of deformed wing virus. When you start to look at what's going on in dead and deformed bees, that's where you see this quantity change. So there's a lot of deformed wing virus in these bees that are demonstrating symptoms. So that's the bees, and then the mites collected from those bees. So again, there is evidence that you have something like deformed wing virus present in a colony. It's at very low levels without the symptoms going on, right? So it's really this abundance change that gives rise to the symptoms. Okay. One of the things that I think is really interesting about viruses with respect to something like Varroa mite is that there's evidence that the transmission route also impacts pathogenicity. So how many viral particles does it take to cause symptoms? So like I just showed you here, the amount of virus you have is related to whether or not you show symptoms. But in addition to that, how you acquire the virus may put you somewhere on, may put you in a different category in terms of showing those symptoms. So just to give you an example to clarify that, so Varroa mite, as a vector of a virus, transmits the virus with high pathogenicity. So if you get the virus from a Varroa mite versus some other route, you tend to be more likely to show symptoms. So So here I have acute bee paralysis virus as just an example. You can get this virus multiple ways. You can get it from feeding with another worker bee that has it, or you can get it from a Varroa mite. If you get it from worker feeding, it takes about 1 billion viral particles, those tiny little virus packages, to kill a bee, to kill you. If you get it from Varroa mite, you only need 100 particles to lead to your death. So this has to do with how the virus gets inside the body. So remember, how does it make you sick? If you get it through your digestive system, that could be very different from a mainline delivery from a Varroa mite. So one of the take-homes here that I think is really clear is Varroa, again, is a sort of gift that keeps on giving. But if you have these present in your hives, not only could they transmit viruses, but it's a particularly deadly form of transmission. Okay. Another thing to consider here is that there are a lot of different factors that impact symptom expression. Because again, we're not looking at a vaccine or an easy way to get rid of the virus themselves, but we're trying to manage the symptoms. So how can we do that? One way to do that is with nutrition and diet. So there are some interesting studies, new studies showing that phytochemicals, these are chemicals produced by plants that are naturally present in pollen and nectar. Some of these chemicals have been shown to reduce deformed wing virus abundance. So when you feed the bees these particular chemicals, they tend to show lower levels of deformed wing virus, less likely to show symptoms. Something like diet also impacts the health and immune function of a bee, generally speaking, which means it'll be more resilient to an infection and more likely to overcome it. I put this picture up here. This is from a paper showing that gut structure in the honeybee. This is the midgut, so part of the digestive system of the bee. The structure of this gut is strongly influenced by whether bees are actually feeding on honey or sucrose. So these different pictures are showing bees that were fed just sucrose, a mixture of sucrose and honey, and that were fed fully on honey. These are adult bees. And there's nothing inside these guts. This is just showing their size and morphology. And you can see that when bees are eating just sucrose and not having any honey, they have these very fragile guts that are actually smaller. So the measurements show that. Honey-fed guts are healthier. Certain of the viruses that bees interact with, one example is that Lake Sinai virus, one of the Lake Sinai viruses. That is a virus that's primarily found in the gut. You can easily imagine that a diet that results in a fragile gut is also going to be more susceptible and show greater symptomology in response to that virus. So diet plays an important role here in terms of symptom mitigation in complex ways that honestly we just don't know about yet. There are other factors that impact symptom expression of a virus. So this was an interesting study because they were actually looking at what is considered to be an inert part of a pesticide, the adjuvant. So this is the kind of vehicle that they use to put the active ingredient of a pesticide before they spray it. And what this study showed is that if you experience this adjuvant alone, which was called OSS, it doesn't impact your mortality much. So they treated bees, they look at the ones that survive over time. So if you're dropping off really quickly, it suggests greater mortality. So here's control, the red. These are bees that haven't experienced anything. These are the ones in green that have experienced just this inert part of the pesticide. They're also fine. If you infect the bees with a virus, you can see it impacts their survival. And then this orange line with the lowest survivorship are individuals that received this adjuvant and the virus, okay? So in this case, this is called an interaction. There's an interaction between this chemical product and viral infection. So again, here, if you're interested in improving the longevity of your bees, you can avoid exposure to these chemicals, you can avoid viral infection, but hopefully you avoid having both of those stressors interacting to impact mortality. They looked at a bunch of different, they looked at a viral cocktail with 3 different viruses in it. And they showed that one of these viruses in particular, the black queen cell virus, was the one that seemed to be impacted directly by this pesticide adjuvant. Okay, so this is a great example of how these stressors act together. Okay, this is an interesting feature of viruses as well. So I mentioned several times that the abundance of the virus impacts symptoms. So if there's more virus, more likely to show symptoms. That relationship changes across different viruses. So this is a graph that I made, basically looking at risk of mortality or symptoms when you're infected with a virus, 3 different viruses, and then I colored them based on their likelihood of showing symptoms. So for example, the deformed wing virus, even at this very high abundance, is only kind of a middle likelihood of showing symptoms. This is chronic bee paralysis virus at relatively low levels, very unlikely to show any symptoms. LSV2 has higher levels than this chronic bee paralysis virus, lower than DWV, and highly likely to show symptoms. So this is based on studies where people have looked at the level of the virus and then looked at whether symptoms are likely to be expressed. So this is just to say, some viruses are more deadly than others, which hopefully is intuitive. We don't all know all the details about that. But this is a place where knowing which viruses are inside your colony could be very helpful. Because if, let's say, you have high levels of a certain virus that's going to show symptoms, you may be able to counteract that, whereas others you can ignore and not worry about as much. Okay. As I mentioned, Varroa plays an important role in impacting transfer of viruses and also symptomology. But there's also a relationship in the other direction. So virus infection benefits Varroa mite production, which is terrible, but it's another example. So mites tend to reproduce better on bees that are infected with the foreign-wing virus. So as many of you know, the mites actually reproduce by feeding on pupal bees as they develop into adults. Well, if this pupa is infected with the foreign-wing virus, more mites are produced during this process. It's not known why that's true. It's possible that the mites are diminishing wound healings. They're just basically stressing the immune system or suppressing it in some other way that facilitates mite feeding. So again, these are interacting factors. I wanted to make some comments about the impacts of viruses kind of generally speaking. Viruses have been associated with colony collapse disorder in a few studies. This is a really early study from 2007 looking at viruses in colony collapse disorder bee colonies. This particular study implicated IAPV, a specific virus. Subsequent studies have not always implicated that virus in CCD, and in general, the state of the knowledge in this area is that the cause of CCD specifically is not known. But one pattern that does seem clear is that there are often cases where you have multiple viruses infecting colonies with colony collapse disorder. They also tend to be infected with multiple pathogens in general, including things like Nosema. So this is an interesting case that, one virus of any given type a colony may be able to tolerate, but when you get multiple viruses, you start to see these non-additive effects leading to something like CCD. This was another study that looked at colony collapse disorder and again emphasized its role for co-infection, meaning multiple viruses at once. So they found that CCD colonies were more likely to have multiple viruses or more likely to have multiple Nosema species. If you add all of the pathogenic organisms, again that's bacterial pathogens, fungal, and viral, you find that there are more in these collapsing colonies. So no definitive single cause of CCD, but when you have a bunch of pathogens, it increases your risk. So that was colony collapse disorder, which some of you may know is a very specific syndrome of symptoms that honeybee hives display when they die. I'm not going to go into a lot of detail there, but there are other causes of mortality that are not CCD. So not every hive dies from colony collapse disorder. But interestingly, these factors that are associated with higher rates of CCD are also associated with higher rates of general mortality. So if we can kind of solve one problem, we can solve multiple problems. So this is a pie chart from the Bee-Informed Partnership, self-reported causes of hive death in 2015-2016. So this is from a survey of beekeepers nationwide. And you can see there are these big chunks, Varroa mite and other, queen failure and other. So what this means is that there are mites present, but then other things going on as well. That's a twice as big piece of the pie as just Varroa alone. So again, it's viruses, other pathogens, other general stressors playing a role. I wanted to show you some data from Kentucky. So this is data that's part of the sample of bees that Tammy Horn has been collecting from beekeepers across the state. To be eligible for this collection, you have to have at least 8 colonies. And this is basically a pooled measurement of the viruses that have been observed. So whether they're observed in these hive collections or not, and it's relative to a national average here. So the blue is Kentucky and the black is national. And these data, by the way, are freely available online. So what we see here, there are a couple things that are interesting to me about this. So we have high levels of deformed wing virus or high prevalence, just like everybody else, basically. There are certain viruses that don't seem to be very prevalent in Kentucky relative to other places. We have others that Kentucky seems to show exceptionally high prevalence. This LSV2 is particularly interesting to me. This was the one that I mentioned predominantly occurs in the gut. And I'm very curious in developing a study right now looking at how nutrition impacts viral abundance for LSV2 specifically, since it seems to be excessively present here. So there are data here that are missing if you think about what we need to know. So for example, we don't know much about how viral titers and abundance change over the course of the season. That if we knew that information, we may have a better idea of the stressors that change symptomology or viral abundance. So these data, this survey was designed to get a nationally representative sample, right? So we have some Kentucky data, but the survey was designed to target different areas of the country and give a big picture. If we want to know what's going on in Kentucky, we need much what's called finer grain sampling. So think about it like if you wanted to get a sense of, on a national level, who's going to win the election versus a local or state level. It's a very different sampling strategy. Talk about LSV2. Okay, so I wanted to use that to segue into a new project that we are trying to get going in the lab, hopefully with some funding from the Kentucky Ag Development Board, where we would like to develop, if we can, a sustainable viral testing service for beekeepers in Kentucky. The goal of this is to generate a report that looks like this. I know that you can't really read it, but this is the idea. You could get a hive health report for your honeybee hive, a specific individual hive. It will tell you about presence or absence of different viruses in those hives relative to the national average and what's typical for Kentucky. We would like to also provide viral abundance data. This is not easy data to get, and for that reason it's difficult to even send samples anywhere and get this information. But it's really important for understanding the risk of mortality or the risk of showing disease symptoms. So that's our goal there. And here we could also easily provide mite counts. So what we have here is a recommendation for how to treat this colony given its viral profile and whether or not it may show symptoms. So here a beekeeper can say, what's the likelihood that this hive is actually impacted by the viruses it's infected with, and what can I do to minimize those impacts? So maybe I need to be feeding these bees really aggressively through the fall, where other colonies that aren't impacted by the virus in the same way, you can save your money and not bother feeding those bees. So it's the kind of thing we're trying to do. So please stay tuned on that, especially if you're interested in having any sort of viral testing for your hives. We are hoping to roll out the first part of this very soon, which will involve recruiting beekeepers and having them donate bees so that we can get this study, these assays up and running. I wanted to end by making some points about viruses in general for those of you who may have conservation interests, for example. So I've been talking about honeybee viruses here, but what's interesting is that these aren't just honeybee viruses, they're sort of bee viruses more generally. So a lot of these viruses probably started out or through honeybee management, since we know a lot about honeybees, we've managed them for a long time. But these viruses have been found across a lot of different wild bees. So we have different types of mason bees represented here, ground nesting bees, bumblebees of various types, carpenter bees, other types of solitary bees. And many of these show some of these same honeybee viruses. So in this table, each column here is a different virus, important really which one for the sake of this talk. And then the colored in square indicates that virus has either been at least found to be present. The orange was that it was observed replicating, so making new viruses and potentially using the host cells in that way. The red means the virus was pathogenic, which means it was causing symptoms. So honeybee viruses are impacting a lot of other things besides just honeybees. And there's even some evidence that this is because viruses are transmitted on pollen. This is important maybe if you're concerned about transmitting viruses across your colonies. So it is present in pollen balls. Basically, this study collected honeybee foragers. They looked at the proportion that had different viruses, and then they looked at what was going on in their pollen pellets, and they found in many cases that there were viruses present. So it is possible that bees are transmitting viruses to one another by visiting the same flowers and depositing infected pollen and picking it up. But you know, you may also want to consider this for a route of transmission among honeybee hives.
Claire, not that the pollen contains the virus.
That's right. It's not inside the pollen cells. It is on the surface of the pollen. Yeah. So it's another route of transmission. And one question, you know, how infectious are the viruses? I don't know that anybody knows that. Yeah. But certainly enough that they're jumping species. It's kind of interesting. We have plenty of time for questions. I guess I'll give you a couple take-home messages first. Viruses are cellular parasites. It kind of helps me to understand the biology of how these things are working. So they're getting inside, they're taking advantage of the cell. There is this idea of pathogenicity, so the amount of virus you have affects whether you're going to show symptoms. Very important that pathogenicity depends on abundance, but also mode of transmission, right? So mite-transmitted viruses are some of the most deadly for that reason. So varroa mites are common viral vectors, meaning they spread viruses, and they increase pathogenicity. So control your mites. You'll hear that, you know, till you're blue in the face, but it's, you know, it's important. Most known bee viruses come from honeybees, but they can be transmitted to other bees. So it's important for us to keep in mind from a conservation standpoint. And some acknowledgements, but I can definitely take questions for anyone who's got anything.
So, go ahead. So there's no cure for none of this.
Yeah, so I don't think that anyone is even looked at, let's say you have a certain viral abundance in a colony. If you do some kind of intervention, let's say nutritional intervention to try to improve the health of those bees, if you track the viral abundance, does it go down? That kind of data doesn't really exist. So, at least not broadly speaking. So I don't know that we can say for sure that we don't have, that there's nothing that can be done, or that we don't know what to do. And that's kind of where we need more data to answer that question. But there's no out and out cure or vaccine for these things. Yeah.
Now, I'd like to say this is groundbreaking for Kentucky. And this is groundbreaking, your program that you're gonna do, because it's not, I don't think it's in very many other states. And you guys should really participate in their program. I mean, it will make, in the long run, your bees more survivable. And especially